In this chapter, we're going to investigate proteins, and of the four different types of biomolecules that we look at, proteins tend to be my favorite. So just as a reminder, the four types are carbohydrates, lipids, proteins, and then DNA and RNA. And we're actually going to look at proteins over two chapters because one chapter is dedicated to enzymes, and enzymes are a type of protein. So one of the things that I love about proteins is that they do things. They're what I fondly call here the workers of the cell. And so when we talk about the structure, these are the reasons that our body functions, is because proteins do the work. Now, when we think of proteins, we also may think of different foods. And so you can see here, I've got some kind of, when I just Google search protein-rich foods, these foods came up. And when we talk about proteins, proteins are going to be, that are found in these foods, or when we talk about protein-rich foods, are the type of proteins that we're also talking about. Now, the name protein is derived from the Greek proteos, which means of first importance. And I think that's really appropriate for the most important of all biological compounds. It's just me. Proteins are really indispensable components of all living things, and they play a really crucial role in biological processes. And so in this chapter, we're going to start with a discussion of the structure and chemistry of common amino acids. And amino acids are just the building block of proteins. And then we're going to progress to show how combinations of amino acids form large molecules called peptides, and then larger molecules called proteins. So without further ado, let's jump into amino acids. So, all proteins, regardless of their origin or biological function, are structurally similar. They're polymers, and poly just means many, of chains of amino acids. So proteins are polymers of chains of amino acids. So a lot of times we refer to amino acids as the building block of proteins. Okay. Now, so let's talk about the structure of amino acids just kind of really quickly. As the name implies, an amino acid is an organic compound that contains both an amino group and a carboxylate group in its ionic form. And so if this was not protonated, that would be called a carboxylic acid. And so that's where the amine and the acid come from. Now, there's hundreds of different amino acids, both synthetic and naturally occurring, but what we know is we have only 20 amino acids that are commonly found in natural proteins. Okay, so we've got 20 common amino acids. The amino acids in proteins are also called alpha amino acids. And many times that alpha is abbreviated like that, like alpha amino acids. You'll see it like that. And the reason is they're called this because the amino group is attached to the alpha carbon. And so if we look up here, this is considered here in black to be the alpha carbon. The alpha carbon is just the carbon next to the carboxylate group. So it's this carbon right next to the carboxylate group. So each amino acid is going to have a characteristic side chain. And I'm going to abbreviate amino acid as AA. Right? And so these side chains are generally called R groups. Right? And those R groups are attached to the alpha carbon. And so you can see here, this would be an R group. It'd be a different, what we call a side chain, and it's different for every amino acid, and it's attached here to the alpha carbon. Now, this R group is what imparts chemical individuality onto the molecule. So it's what controls or is a great deal or part of the molecule's reactivity. And so these R side chains contain different structural features like aromatic rings, alcohols, amino, and carboxylate groups. And we're going to look at those more in depth in just a second. So this variety of side chains causes differences in the property of the individual amino acids and the proteins containing the different combinations of them. Now, each amino acid is going to have a name. Right? A 3-letter abbreviation. And a 1-letter abbreviation. So let's look at these 20 common amino acids. And before we do that, I want to talk to you about amino acids and R groups. I thought we were jumping right into all the different amino acids. But before that, we're going to talk about amino acids and their R groups. So the polarity of the R group is very, very important. And so amino acids are classified based on the polarity of the R groups. And so some of the divisions that we have, you can have neutral or nonpolar side chains. And these generally have just carbons and hydrogens. So a really good example is like CH3. You can have neutral. So neutral just means there's no charge. So I said or here, I meant and. So neutral and polar side chains. An example of that would be like CH2OH. Okay. Then you can have basic side chains. And these tend to have an amine on them. So for example, CH2, 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 NH3 plus. And then you can have acidic side chains. And for example, it would be like CH2COO minus, right? So now let's go forth and look at some different amino acids. And when we talk about amino acids, and we're going to look at these different categories, again, they all have this kind of similar backbone where they have an amine group, an alpha carbon, a carboxylate, and then here are the R groups that you see here. They've got a name, a 3-letter code, and a 1-letter code. And sometimes those make a lot of sense, like cysteine here makes a lot of sense. But glutamine, the 1-letter code here is a Q. Asparagine, the 1-letter code here is an N, because there are other amino acids start with other different letters. So sometimes it makes sense, and sometimes it's a little more challenging. So let's jump into looking at our neutral nonpolar side chains. So when we look, these are amino acids that have neutral nonpolar side chains. And I want you to notice the side chains, and I'm going to go through and just circle them. Proline has a ring there. They almost all contain only carbon and hydrogen. And of course the exception there is methionine, which has sulfur, but that sulfur is pretty well protected by the carbons and hydrogens there. And so this is what makes them nonpolar, is they contain only those carbons and hydrogens. Now, we can also have polar side chains where there's neutral, there's no charge here. And then when we look at these, right, here are those side chains. I want you to notice that these have a lot of carbons and hydrogens in them, but at the end of the chain, there's no charge, and you see that there is some sort of electronegative atom. So we're going to have an oxygen that's bonded to a hydrogen. We're going to have a nitrogen in there that's bonded to 1 hydrogens or 2 hydrogens, and we're going to have a sulfur. And so those molecules there are the clue that, hey, look, these guys are polar. Okay. Now, we can have polar basic side chains. And what does this mean? This means that there's going to be a charge, and these all are going to have nitrogen. So all 3 of these are the 3 basic polar side chains. They have nitrogen and a charge. Okay? And so you can see the charge is here, the charge is there, and the charge is there. And it's a charge on their side chain. I'm not talking about up here. Then the acidic ones over here, there are 2 acidic ones. They're going to have the carboxylate group. From a deprotonated carboxylic acid. So that's a carboxylic acid that has lost its proton. And it's also going to have a negative charge there. And so those are called the acidic ones. Now, let's talk about the stereochemistry that's associated with these amino acids, because one of the things to notice is we have a carbon here and it has 4 different groups attached. And so that means then that it is chiral. So with the exception of glycine. Glycine is achiral. So when we look here, glycine has 2 hydrogens, and so this guy is achiral. But every other of the 19 of the 20 amino acids are going to have 4 different groups, and so they're going to be chiral. And they exist as 2 enantiomers. They exist as an L and a D enantiomer. And how we can tell is in this particular structure, the way it's drawn, is the D is on the right, and here for the L, the NH3 there is on the left. So for the L1, the NH3 plus is on the left, and for the D, this NH3 plus is on the right. And I really emphasize the L particularly because the amino acids in living systems exist in that L form. And so it's really helpful to be able to see that. Now, we have charges here. And one of the interesting things about amino acids is they can form a Zwitter ion. So up until this point, the structural formulas that we've shown have been amino acids that are ionized. And remember, that just means they have a charge. So this is a non-ionized form. There's no charge. And this is an ionized form, and we have the charges here. So this would be our ionized form. Now, amino acids are white crystalline solids, and they have relatively high melting points and high water solubilities. And so all of that suggests they exist in an ionic form. And all an ionic form is a form that has charges. So studies have shown that this is true in both the solid state and in aqueous solutions. Now, the presence of the carboxyl group here and the basic amino group here makes it possible to transfer a hydrogen ion in a kind of internal acid-base reaction, and that product then is called a Zwitter ion. So a Zwitter ion is just a dipolar ion that carries both a positive and a negative charge as a result of an internal acid-base reaction in an amino acid molecule. So one of the interesting things about this then, right, is that the structure of the amino acid in solution varies with the pH of the solution. So the structure of the amino acid in solution varies with the pH of the solution. So what do I mean by that? So for example, let's say if the pH of the solution is lowered by adding some source of H3O plus, like HCl, okay? So we've got H3O plus being added here, and this is an acid, okay? What happens is the pH of the solution is lowered. Okay, and then when the pH of the solution is lowered, the carboxylate group of the Zwitter ion picks up a proton to form COOH, and that negative charge goes away, and now you've got a positive net charge on that molecule. Now, conversely, right, if the pH of the solution is increased by adding a base, so this would increase the pH of the solution, right, the NH2 here loses a proton. And I just want to emphasize that a proton is just H+. So I know this is a flashback to acid-base chemistry, but that's what a proton is, right? And so this is a base, we've added it, and now, right, it's reacted with the proton from here to form water. But look, this is no longer charged, that NH2 is no longer charged, and so now you have a net negative charge on the entire molecule. Now, changes in pH can also affect the acidic and basic side chains and those proteins in which they occur. So all the amino acids exist in solution and ionic forms, but the actual form and charge is determined by the pH of the solution. Now, each of the amino acids, right, assume a positively charged form in acidic solutions and a negatively charged form in basic solutions. So it seems reasonable then to assume that at some pH between the acidic and basic extremes, the amino acid will have no net charge and be a Zwitter ion. Now, that assumption is 100% correct, And the pH at which the Zwitter ion forms is called the isoelectric point. So the isoelectric point is just the characteristic solution pH at which an amino acid has a net charge of 0. Okay, so each amino acid has a unique and characteristic isoelectric point. So those with neutral R groups are near a pH of 6. So this would be a neutral, right here, a neutral R group on that kind of first couple slides that we talked about. So for example, glycine is a good example of this, and its specific isoelectric point, an isoelectric point is abbreviated as a PI, like that, is 6.06, okay? And then what you're gonna see is acidic amino acids are gonna have lower values. So the two acidic amino acids are aspartate and glutamate. And so for example, I meant to tell you, aspartate is also known as aspartic acid. So there's two names for that, and we'll go over that again in class. But I wanted to mention it here as well. And that has a isoelectric point of 2.98. So it's much lower than 6. And then if you have basic amino acids, like lysine is a basic amino acid, then it's got an isoelectric point that equals 9.47, and so it's much greater than 6. Now, my tell on these kind of PowerPoints should sometimes be if I type things, it's because I definitely want to make sure that you have them. At pH values above the isoelectric point, the amino acid has a net negative value. And then at pH values below the isoelectric point, the amino acid has a net positive value. Well, what does this mean? Well, let's look at our general amino acid. We've got H3N plus, and then CHR, and then C double bond O, O minus, right? And let's say this is got an isoelectric point equal to 6, and it's sitting in pH equal 6, okay? And now we're going to come along and we're going to say, all right, hey, look, we are going to add OH minus, right? And that is going to bring the pH to 9. So it is above the isoelectric point. Somebody knocked on my door, and so if it's a little disconnected there, that's what's going on. So we were talking about what do we do if the pH has increased. So at pH values above the isoelectric point, we are going to see that there is a net negative value. And so just like on the previous slide, right, what this means is that the H here will be lost, and now it'll be neutral, and then the carboxylate here will still have a negative charge. Then so that's an example of this first one here, okay? Then if something happens and we add H3O plus, and now the pH value would be really low, let's say the pH equals 3, we are going to see that we've got a net positive value. And what would happen here is the H3N stays charged. You've got CH, you have R, and then we add an OH. So we've added the hydrogen here, and now that's that's protonated. And so that would be an example of this situation. So those are the two examples that we're going to see. Now, if you remember back to General Chemistry 1, amino acid solutions can act as buffers because they react with both H3O plus and OH minus. So they're reacting both with an acid and base. And just as a reminder, a buffer just maintains the pH, so it's not going to vary very much. Now, this brings us to the end of our discussion about amino acids and Zwitter ions. And in the next screencast, we are going to look at the reactions of amino acids as well as peptide formation. As always, if you have any questions, please feel free to stop by my office.